Hello everyone, this is MEENU AGRAWAL coming back to this topic cell. Now we are going to discuss about cell wall. So let's start with your bacterial cell wall. Okay, so we are going to start with bacterial cell wall. You know that bacteria has got a cell wall which is made up of peptidoglycan. So let's look at it. Has got a membrane. Okay, so this is your cell membrane and outer to it is your wall. Outer to it is present is your wall. So this is your cell wall which I am trying to make for you. So this is your cell wall. Okay, so it's closely pressed to the membrane. So this is your cell wall. So you can see the red one is your cell, and there is an and you know the cell wall is made up of peptidoglycan. It's nothing new, I'm telling you. It's your cell membrane. This is your cell wall. Okay, now you know there are various functions of the cell wall. So like if you look at the functions of the cell wall, what are the various functions? It gives shape to the cell. It gives what? Protection to the cell. And it protects the cell from, it gives you can say from bursting when you keep them in hypertonic solution. So various roles is played by your cell wall. It gives an outer to the cell wall there is another layer, okay, which we call as a glycocalyx. Okay, so now if you look at this layer, this layer is of glycoprotein coat. Glycoprotein, if I say, means that this layer, and I told you in that when I told you about prokaryotes and eukaryotes, that it may be present and it may not be present. Okay, so this coat is what? It's called as glycocalyx. Now this is made up of what? Glycoprotein. Protein. That is, it is made up of carbohydrate and protein. So this is made up of glycoprotein coat and it is called as glycocalyx. And this glycocalyx layer can be, it is of two types. What is that? It could be slimy or it could be your capsular. So it depends. Some bacteria have got slimy and it gives different property. Some pathogenicity property to the bacteria, gives different property to them. And this layer and these all three layers, all three layers. That is your cell membrane. If I look at your cell membrane, cell wall, and your glycocalyx layer is together making up your what? Cell envelope. Okay, in case of your this bacterial cell. Okay, so they are making your cell envelope. Fine. Now it's not that ki glycocalyx, no, we are talking about glycocalyx. It's not present in eukaryotes. It's not there. It is also present in eukaryotes. Like you know that RBC is coated by a glycoprotein coat, which is giving then the property whether it is A, B or C. So this glycoprotein is also present, in eukaryotes. Karyotic cells also it's there. Okay, in animal cells, although animal cells they don't have a wall, but they have this glycocalyx layer which is made up of glycoprotein, which gives them different property as I told you. Okay, so now moving on with the plant. You know about plants, so let us talk about plants. So plants have got plants have got cell wall, you know uh, and their cell wall, let's talk about, it is made up of cellulose. All of you know that, isn't it? Now if you look at the cell wall of a plant cell, then what do you find? That it has got two type of cell walls, isn't it? It has got primary and secondary cell wall. Primary cell wall is made up of cellulose, okay, and it is present in all the plant cells, whereas secondary wall now is present in some of the cells and may not be present in some of the cells. Now let's, if I have to draw for you, then just a moment. Like if I have this primary, now there is a middle lamellae, okay, and around it what will happen, your primary wall will be formed, okay, your primary wall will be formed, and which is a thin wall, okay, which is a thin wall, okay. So I will try to make it thin. So it's a thin wall, it's a thin wall, okay, it's a thin wall, and which is formed around this middle laminae, both the sides. The if the cells are there, then this is the primary wall, okay, this is your primary wall, okay. So this is the primary wall which is formed, okay, and once, and it is flake, and it is it becomes rigid later on, okay. Then the cellulose provides what it provides. If I talk about primary wall, then if I have to talk about it, it has got various roles. It gives what, it gives shapes to the cell, okay, it gives what, it gives shapes to your cell. It gives what, it gives them protection from bursting, it prevents them from bursting. Give them certain kind of rigidity to the cell or turgidity to the cell. And turgidity basically is because of the water that is present there, which is giving, but it is pushing on the wall, okay. So somewhere you can say it gives rigidity and turgidity, turgid and rigidity to the cells, okay. So it gives various uh, imparts various character to the cell. And now inner to this cell wall, this is your cell wall, this is your primary cell wall, okay, this is your primary cell wall, okay, this is your primary cell wall, and inner to this primary wall is basically formed your secondary wall, okay. There is deposition of certain substances. There will be what, deposition, there will be what, there will be deposition of certain substances. Like you must have heard about lignin gets deposited in case of your xylem. So there is what, deposition, there is deposition of certain substances, so they get deposited, okay. And this layer is quite thick, okay, secondary wall. It could be suberin deposition, it could be lignin deposition, and the areas where they are not deposited, you can see they create the area which is called as pits, through which you know about the cell wall. If you look at the property of the primary cell wall, it is what, it is permeable, it is what, it is permeable, it is permeable, okay, it is freely permeable, okay. The substances therefore can move, through roots. If you soak a seed, it is able to take up water. Why? If it is permeable, it is permeable, that's why. Now due to this deposition, the secondary deposition, what is happening now, which is making the wall impermeable, which is making it, suppose this is lignin, hmm? Lignin is getting deposited here, then it makes the wall what, impermeable. Therefore you find that through these pitted areas where this secondary wall is not present, through those area water can move out or can move in. Okay, so this is about the secondary wall in case of your plant cell. So this is about plant cell wall. And remember, secondary cell wall is always now inner to the primary wall. It's not outside, it's inside to the primary wall, okay. And now moving, I'm showing you what are the different kind of connections that the cells have, okay. Now you can see here that this is the cell wall, it's clearly visible to you people. Okay, the green one is your wall. No, this is your cell wall. Okay, now you can see here there is a cytoplasmic connection. Okay, this is a cytoplasmic connection in the cell wall of plants. Okay, so primary wall is not present there. Okay, so they have a cytoplasmic connection, and this connection is called as plasmodesmata, through which what is happening, your bigger organelles cannot move, but smaller ions, now molecules can move through it. Okay, organelles are bigger in size, they cannot move from one cytoplasm to another cytoplasm of the cell, but smaller molecules can move, and this is how they communicate with one another. One cell is communicating with another cell. Now this was about your plant cell. Okay, so remember, plant cells have got plasmodesmata connection. This is plasmodesmata connection. Okay, they are having, this is a cytoplasmic connection. And now coming back to this animal, this is a tight junction. Okay, they just seal the two membranes. Okay, they just seal the two membranes and nothing is able to move. Like there are now surfaces in our body, like if you take urinary bladder, if it had been leaky, then the urine will move to the other cells, the extracellular fluid. So to avoid it to move out, these junctions are what? Tight. They won't allow anything to leak out of them. Okay, so such junctions are called as what? Tight junctions. Okay, now if you look at here this is a junction which is sealing, which is bringing, which is holding the two membranes together. Okay, so these are the desmosomes. As you can see here, I am just using the pointer. So this is the desmosomes, which is now coming together and interlocking among themselves in such a way that the two cells are hold together, they are weld together. Like if your skin cells ना, अगर ये आपस में जुड़े नहीं रहते तो would they have been at the position? No, no. So this is how these cells they hold together and themselves. Okay, and they give that particular shape, that structure. जैसे कैसा होता है कि बिल्डिंग बनाते समय आप क्या लगाते हैं, प्लास्टर करते हैं ना, सीमेंट लगाते हैं to hold them at their positions. So same things they are also doing here. And the third one you can see here is a gap junction or a gap channel like है जिसके through your ions, your smaller solute molecules है ना, glucose and all can pass through. Okay, so that is how the one cell is communicating with another cell, passing certain other kind of substances which need to be passed on from one cell to another cell. So this is how the some small molecules which can act as an information molecules also, they are able to pass through these gap junctions even within basically we are talking about one cell how is connected with another cell. So these are the different kind of junctions in animals. Okay, there are many but very few but basic ones I have discussed with you. Okay, so with this cell wall is complete. Goodbye, God bless you.